Western blotting, it is another technique that is used for the analysis of immobilized proteins. So this is not used for nucleic acid, rather it is used for proteins. So protein molecule from any tissue, they can also be separated by using polyacrylamide gel electrophoresis, and after separation then these proteins on the membranes, they can be detected by using antibodies. Now antibodies, they are proteins from the immune system that are highly specific in their action, so that a specific antibody can react with a specific antigen or protein molecule. So here we also immobilize protein on a nitrocellulose membrane or nylon membrane, and after immobilization then we use antibodies to detect the fragment of proteins. This diagram indicates the overview of Western blotting. So in the first step, different protein molecules like the DNA or RNA, they are separated by using polyacrylamide gel electrophoresis, and from the gel then the molecules, they are immobilized or blotted on the nitrocellulose membrane, and then in the third step the proteins then they are detected by using specific antibodies, and finally they are detected. This is another diagram that indicated detection of proteins. So here protein has been immobilized on the nitrocellulose membrane, and then the primary antibody, it is used for the detection, and then this primary antibody, it is detected with another antibody that is radio labeled, and this antibody, that is secondary antibody, it can be detected by using specific enzyme. When it will react with secondary antibody, then it can produce color that will be detected. So it is a sort of sandwich technique where the primary antibody, it is reacting with the antigen, and then the secondary antibody, it will react with the primary antibody, and the secondary antibody, it is actually labeled with some sort of radio labeled material or any non-radioactive material that is finally detected. So this is another approach which is called as sandwich method, where we use two antibodies for the detection of target proteins.